Okay, the proteins, they are the macromolecules or biomolecules which are mainly made up of elements which are carbon, hydrogen, oxygen, and nitrogen. And moreover, they have some
Sulfate.
sulfur molecules also, sulfur compounds also, or actually, you know, we will be discussing this thing once we will go to that disulfide linkages.
Okay.
Okay, now the point is what we know, proteins are mainly made up of carbon, hydrogen, oxygen, and nitrogen. So nitrogen is addition as compared to carbon, hydrogen, oxygen, which were there in carbohydrates and fats. Correct? Okay.
Yes.
Now, first of all, once we go for the structure, what is the importance of the protein? What importance the protein they are having? Proteins, they have structural as well as functional importance. You know it, yes? Proteins, they have structural as well as functional importance. Now, what is the structural importance of the protein? Protein, they are made up of amino acid, obviously, and they are fibrous protein. They make up your structure, body. Fibrous proteins.
Tissue.
Or tissue, obviously, tissue. They actually make up.
Enzymes.
No, enzymes, they are responsible for, you know, they have physiological importance because they do some other kind of work. They are globular proteins. So one protein, they are the part of the structure, which we will be discussing later on, that they are structural protein, mean fibrous protein. They make fiber. Okay, tissue fiber. And some protein, they have their functional importance. They are actually, you can say, in the liquid form or some kind of a liquid form, like enzymes. Okay, and they have their physiological, functional importance, other functional importance, like enzymes. They are globular protein. We will be discussing this thing later. Okay, so mainly once we are talking about this sulfur is also one of the integral part of the protein. We will be discussing that thing a little bit later. Okay, now next we are moving. Now, once we are talking about ribosomes, so ribosomes, they are actually what? DNA transcribe messenger. This already, we learned this thing, I think in O level, you know it, correct? Yes. Transcription, translation, and then polypeptide chain is made, and this is the protein which is made. Okay. Even what are ribosomes? These are the cell organelles which are responsible to make peptide bonds, or which are responsible to make polypeptides. Isn't it? I'm just I'm just skimming through with this. Now, actually, we should know this part. So amino acids, they are the basic structural unit of proteins. Many amino acids, they combine together to make proteins, correct?
Yes.
Now, once we are talking about these amino acids, okay, what we need to know about amino acids here. Amino acids, they have one alpha carbon. This, which is this pink color carbon. Okay, they have one central carbon atom. Okay, this carbon. This is alpha carbon. So on that alpha carbon, then obviously carbon will be having 4 bonds. Yes, single bonds on both sides. Yes, so it will be having one here, one on this side, one on this side, and one on this, correct? Now, amino acids, these are the biomolecules which have the properties of both acids and base. They have 2 groups. Acids and bases, alkalis. Okay. Correct? So here, what will be here? It will be here, we will be calling it, this will be COOH, correct? Carboxylic. Carboxylic bond on one side, correct? Yes. And the other side, it will be having amine group. NH2. Very good. It will be NH and 2 is there. 2 is not written like this, but what to do? I cannot do it now. On top, there is actually here in the picture, which is on top left, hydrogen. This is actually R, and down we are writing here the hydrogen group. Hydrogen. It's okay. It will be here. Yes, almost this structure and this structure are the same, correct? So on one side we have carboxylic acid group, this COOH. This COOH can be written in this format. Yes, carbon, double bond, oxygen,
Yes.
and OH, correct? And amine group, obviously, it will be written as the way it is written on your top left, correct?
Yes.
Now, this is the alpha carbon. Now, here, amino acids, they are amphoteric compound. Amphoteric compounds Amphoteric compounds are the compounds which contain the properties of both properties of acid and alkalis. Double properties they have. Amphoteric. Shall I write the spelling?
Yes.
Amphoteric compounds.
Which has?
Which have the properties of both acids and alkalis. So both the properties are there in between. So one part. So or otherwise you can say they have one positive part and they have one negative part, negative charge. Once we are talking in terms of charges, yes. So these are the compounds which have both positive and negatively charged ions or compounds. But better you go for the thing which amphoteric compound. These are the compounds which contain both acid and alkalis as their a part of their structure. Okay, amine group is alkalic, alkaline, and carboxylic group is obviously this is the acidic group.
Hmm.
You got it?
Yes.
Okay, next thing is all the amino acids, they have the same integrity. This is the basic structure of all amino acids. There are 20 naturally occurring, we call them essential amino acids. Please you add this thing. There are 20 essential amino acids.
In our
Naturally
body.
occurring and encoded by DNA. So you, shall I, what, sorry?
In our body.
Yeah, in our body, in all living organisms, not in our body, in all living organisms, essential amino acid. We call them essential amino acid. We call them the essential amino acids. Why they are essential amino acid? Because all the living organisms, they contain, their body is made up of these, mainly these 20 amino acids, which are joined together in different forms to give different kind of structure. So how our body is different from other animals, because other animals also they have the same 20 naturally occurring essential amino acid, but their sequence is different, so that way they look different, correct? This first thing.
Yeah.
But basic structure formula of each, so you can say this is a general formula of an amino acid, generalized formula of the amino acid. What is all amino acids have the same general formula? All these three things, 1, 2, and 3, carboxylic group, amine group, and hydrogen. It is common with them. So these 20 amino acids, they will be having this structure common. Only they differ in R group. What is R group? R group is what?
Side chain.
नहीं, R group को क्या कहते हैं chemistry में? What we call R group in chemistry? I don't
हमें भी
have to
सर ने
talk
यही
Urdu.
बताया था साइड चेन.
I'm sorry?
सर ने हमें यही बताया था साइड चेन.
Okay, actually the thing is, this is alkyl group. We call it alkyl group. So all the amino acids, they have the similar basic structure. They are different from one and another through the alkyl group, the R group. Okay. Now, if instead of this R group, if we replace this R group by a simple H, for example, if I take it here, this R group, if I keep it here, H, this you have to write, please, okay, you have to remember this part. This H, if I write, now this amino acid will be named as glycine. This is glycine. So R group, if this R group is H, so this is glycine.
Okay.
Okay, now if this R group is replaced by an another thing like, for example, this R group is replaced by CH3. 3 will be down, please. Okay, this will be under
Hmm.
hydrogen. Yes. So if it is down, CH3, so this amino acid, now this amino acid will be named as alanine. You got it? So if it is, this will be
Yes.
alanine, this amino acid. So obviously once another it will change CH3, CH2, blah, blah, any other structure changes here, it will make another type of amino acids. So this we can see it. Shall I remove this screen now?
Yeah.
Let me remove it. Everything I removed, okay? Now, the point is,
You can also use the whiteboard if you want.
I'm using it as a whiteboard, but I don't know, I mean, once we are sharing the option, yes, in the sharing option, it becomes better, because on top I'm writing, so some kind of references, if I need, I can just directly show
Hmm.
it to you. Okay, comparative.
Okay.
Okay, now if I move next down, I think I have to remove this and then I will be able to, yes. Now look down. So there is leucine. So this is R group. Yes, this is R group. It is different. Now serine, it has CH2OH here. Then this is the most important part. Cysteine. This is the most important thing. CH2, and this is actually R group. I wrote the R group on top. Please remember, okay? Here
Yes.
they are
Yes.
writing the R group down, so it does not make difference. Okay? So please remember, this is acid. Cysteine have sulfur.
Hmm.
Cysteine has sulfur. So you just remember this point. This is really very important. Please make note of this structure. I told you three structures. One structure is glycine, yes. Second structure of amino acid is
Yes.
alanine, and third structure of amino acid that you should be aware of. If you don't know this structure, the whole structure, but you should remember cysteine is the amino acid which contains sulfur.
Hmm.
So here it is very clearly they are mentioning R group determines the chemical properties of any amino acid, correct? So it is determining the different nature of the amino acid that we already, correct?
Yes.
Okay, now next if we go,
Yes.
now they are showing phenylalanine, then glutamine, structure, aspartic acid. So there are different structures. So R group is different everywhere. You see, CH2, CH2. So this is all of them. They are different basic structural unit. Rest of the structure is the same. They are just different from each other through their R group. Is it clear?
Yes.
Okay.
Yes.
Now there are 20 amino acids encoded by the DNA. That already genetic code term, genetic code.
Hmm.
There are three nitrogenous base pairing which code for one particular amino acid. We did it in translation. One messenger
Yes.
RNA is there, so and which is made due to the transcription. During the translation, these ribosome they encode or decode the information from the messenger RNA and bring down. You remember that sequence of bringing their relevant amino acid and making a polypeptide chain?
Hmm.
Yes or no? You are saying yes for no
25%.
reason. Actually, you are not sure actually.
25%.
Okay, 20 is oh, no problem. Even 25% also, if you are remember that part, so just remember there are three nitrogenous bases like A, T, C, or A, if you remember AUG. if you remember AUG, it codes for one amino acid which is known as AUG. It is known as methionine. I will be writing AUG. This is AUG. It codes for, this is the code, genetic code for one amino acid which is known as methionine. This is the starting codon. I don't know you remember or not. You don't remember, yes? But please remember these three nitrogenous bases, okay? Nucleotides, actually, these are nitrogenous bases. They code for one particular amino acid, genetic code. So they were, there were 64 different codes were available.
Yeah.
Oh, and each code for one amino acid, there is one particular kind of, so these kind of things they were there. Now you remember?
Yes.
There was start codon, there was end codon, nonsense codon, that these kind of terms, if you recall, okay? I'm removing this now. Okay, next thing is folding the polypeptide to form. Okay. You just give me a reminder, you know what? You just give me a reminder that I have to tell you about something was there. Yeah, this is, oh no, it will come. Now, finding the polypeptide. So what kind of bonding will be made between the polypeptides? Yes, so two amino acids, they will be joined together with the help of one particular bond that is known as peptide bond. Correct? So this is phenylalanine, glutamine, tyrosine, serine, isoleucine, methionine, phenylalanine, all these, this is the polypeptide chain, yes? And each amino acid is combined together with one bond. This bond is known as peptide bond, correct? Yeah. This peptide bond is this results due to the condensation reaction between two amino acids. This condensation reaction takes place. This condensation reaction takes place between what?
Two amino acids.
It takes place between the amine group and carboxylic.
OH plus H.
Yes, carboxylic group and amine group.
Yes, amino group and carb.
Normally, you know what the primary structure, they are calling it phosphodiester bond, yes?
Yeah.
But anyway, just remember, this will be made in between nitrogen. Nitrogen is involved, yes? And carbon is involved. Peptide bond, please ignore this part, this phosphodiester bond, blah, blah. You just remember peptide bond, okay? Ignore that. Don't write this, okay? Just write a peptide bond. What is a peptide bond? Peptide bond is made between carbon. This will be made between the carbon.
We did it in the class.
Yeah, you did it in the class?
Yes.
So you are two teachers are teaching you biology?
Yes.
Eh?
No,
simultaneously, normally it happens like this, correct? So there is no harm. We were also doing the same thing, like half portion is divided, one teacher is doing one portion and the other teacher is doing the other thing.
One teacher is there, he is teaching us.
Same teacher is teaching both things, yes?
Yes.
In different periods, correct?
Yes. We have period for 1.5 hours.
Okay, 1.5 hours. Okay. Now the point is, once we are talking about it, but you were telling in the this one lipids, phospholipids. You were not into that detail. You still have to discuss this. So that is why I was wondering how come.
One girl asked that we didn't did phospholipids, right? And then sir told yes, we didn't.
So if the that girl is not asking, teacher will never go to the phospholipids, yes?
I don't know.
Okay, anyway, the point is we are for the peptide linkage. Peptide linkage is made between the carboxylic group and amine group of two amino acids.
Yes.
Okay, and this is condensation reaction. The bond which is made, that is a peptide bond, and this result in the formation of a water molecule. Obviously, this is the feature of This is the feature of a condensation reaction, correct?
Yes.
Okay, now I feel you can make this in your book. You have this condensation peptide bond is there, picture is there in your book.
Yes.
You just go through with the structure. I just want to see whether I have. Yes, it is there. Look at this one. This is the peptide linkage which is made. So here on one side, this is hydroxide of carboxyl group and hydrogen of amine group. They combine together, they leave,
Amino.
and carbon and this nitrogen, they are held together by the bond. Here, peptide bond.
Yes.
You will remember this part, correct?
Yes.
Please sketch it. A small structure you can sketch. Make, the way you have made the general structure of tribe. You send me the picture, yes? About
Yes.
this glycosidic, no, this one saturated bond, unsaturated, saturated,
Right.
un, a double bond, yes? Make a picture of like this, a
Yes.
general picture for this. Make two amino acids, okay, general amino acids together, and make a peptide bond in between them and send it to me now.
Okay.
So you find it easy, yes? Hello? Are you done? Yeah, correct. But once you make this thing, Mariam, only one thing which is missing. You always make small mistakes. Plus water molecule that already you indicated, yes?
Oh, okay. Yeah.
And you have to make this thing. And one more thing, please make the arrow. Like this bond is made, so the arrow will be pointing down, okay? From this, where we have made the water molecule, it is pointing down. Then it will be making a peptide bond that exactly that we have made already, correct? And then plus
Yeah.
water molecule.
Okay.
Perfect, perfect. Okay,
Hmm.
okay. Now next thing is Now, structurally, proteins, they have three structures, okay? Structure of a protein. Now, first structure is primary structure of the protein, secondary structure of the protein, tertiary and quaternary.
Okay.
Primary, secondary, tertiary and quaternary.
Okay.
Okay, now once we are saying about the primary structure of the protein, this you'll be writing it down. Primary structure of the protein gives us information about the number of amino acids and their sequence. It gives us information about the number of amino acids and their sequence.
Primary structure.
It gives us information, gives us info about the number and the sequence of amino acids in a protein. Okay, so primary structure gives us information about the number and the sequence of amino acid present in a protein. Okay, this is primary structure. Now, there was one scientist with the name Sanger. I don't know how you pronounce it. It is Sanger. Okay, Sanger. He was the first scientist who actually made the primary structure of insulin. Insulin is made up of 51 amino acids together. 51 amino acids. Okay, so there are two polypeptide chains. One chain is containing 30 and other chain is containing 21 amino acids, so they are held together. Okay, but Sanger found all 51 amino acids which are making insulin.
Okay.
Okay, so this is the primary structure of insulin. Then this hemoglobin is made up of 574. This is very important. Hemoglobin is made up of 574 amino acids. Hemoglobin is made up of 574. Okay, and this hemoglobin is made up of hemoglobin. What's sure? Hemoglobin is made up of 574 amino acids. Okay, and there are 4 polypeptide chains. How many chains? For insulin,
4.
there are 2 polypeptide chains. Correct? One chain contains 21, other chain is containing how many? 30. So 21 plus 30, so it is 51 amino acids together. Correct? Hemoglobin, it is a compact structure. It is made up of 574 amino acids. Okay, they are made up of 4 chains of polypeptide chains. One chain contains a group of chains. So there are 4, yes? Out of those,
Yes.
2 are made up of 141 amino acids. That's it.
2 are.
Okay? And other 2, they are made up of 146 pair. Okay, amino acids. So there are amino acids. So actually there are a couple of pairs of polypeptide. So there are 2 pairs of 141 amino acid, one polypeptide chain, and other is 146 and 146. So double. So this is also double. This is a double, and the next one also the double. Okay, this is also double. This is also double. So 146, 146, 141, 141. So it will be making 574 amino acids. So this is the important thing. All the protein, they are highly specific. You know it, yes? Any change in the sequence of amino acids which result in the mutation, the people who are suffering from sickle cell anemia, sickle cell.
Yeah.
There is only one amino acid change out of 574. Only one amino acid. which is being replaced by another amino acid, and the problem happens. This is a big deal. Okay, so this is primary structure of the protein. It gives us information about the total number and the sequence of the amino acid present in a polypeptide in that particular protein. Correct?
Yeah.
I'm removing it. Secondary structure. So obviously now you already know that all the proteins, they are made up of polypeptide chains. So what is the secondary structure? These polypeptide chains, they are coiled.
They are?
They are coiled. Wait, I will be writing for you. They are coiled around.
Oh, they are folded.
Folded or coiled. Achha,
Okay.
you're using another word. They are coiled or folded. Actually, first what we have to do, they are actually coiled around, okay? And these, since they are coiled around, so obviously, you remember what is the structure of DNA? Structure of the
Yeah.
DNA, there are nitrogenous bases, yes?
Yes.
These nitrogenous bases, they are held together by hydrogen bonds, you remember?
Yes.
So these hydrogen bonds, what is the function of these hydrogen bond? Two strands of DNA, they cannot fall apart. Why? Because they are held together tightly in between A and T, adenine and thymine. There are double bond, and there is between guanine and cytosine. There is a triple bond. You remember this thing, yes? From
Yes.
GCSE. So same is the case here. Same is the case here. These, the polypeptide chains, yes, for example, in the case of insulin, how many polypeptide chains are there? Two. Yes, so these two polypeptide chains, they will be held together tightly with each other, so they cannot get separated. So they are held together by hydrogen bonds. So this will, so technically speaking, they are folded. First fold. So once they are folded, so what is the advantage of folding?
So before this, we don't have to do the disulfide bond and hydrogen bond.
No, yes, no.
Ionic
Folding,
bonds.
they will become compact. You just remember this thing. They will become compact. You understand? They will become compact. So they will be they will not occupy, obviously once you fold something, so they will occupy less space, correct? So we have to compact them.
Yeah, that I know. I'm asking about the disulfide hydrogen bond and ionic bond. Before this, we have to do that, or after this, there will be the bonds?
No, within this you will be knowing it. Hold on. In English, we call it hold your horses. Okay, we'll be reaching to
No,
that disulfide and hydrogen ionic bonds also. Okay.
I'm asking because we did the bonds first, and then we will do the secondary structure and tertiary structure.
No problem. No problem.
And
We are doing it all together. Okay. That disulfide linkage and ionic bonds, we will be doing it all together. Okay. Together we will be doing all of them. Anyway, you understood? Yes. Secondary structure is the folding. You know, one more thing, there is a coiling. Obviously, secondary structure is compact structure. Yes, you have to fold. If you will be folding, then they will be occupying less space, correct? So these foldings, sometimes, you know, there is one term which is known as alpha helix and beta plated.
yeah.
Yeah. So these foldings, have you seen this picture, by the way?
Yes, there is in the book. Pictures in the
That
book.
picture is there in the R group is present where? It is present outside on external side. R group is present externally, okay? Another is like, I'm just quoting from the book, okay? Another is beta plated sheet in which the polypeptide chain folds into regular plates held together by hydrogen bond between amino acids and carboxylic acids of amino acids.
Yeah.
You have this thing with you or no? So there are
Yes, I have.
alpha helix and beta plate. I don't know what you beta pleated. Yes, beta plate. what do you call it? How do you pronounce it? Beta pleated sheet. Yes. So
Yes.
these are the characteristics of secondary structure.
Yeah. These
Okay.
are written in the book.
Yeah. So next is tertiary structure. Now,
Three-dimensional folding of the secondary structure.
yeah, it is tertiary structure is again the secondary structure is further coiled. Yes. Now this
Hmm.
further coiling need disulfide bridge. Now, for example, first the coiling took place. Which hydrogen interaction was there? Which bond was there in the secondary structure? It was. Acha, what is the bond involved in primary structure?
Hydrogen.
In primary structure,
Covalent or hydrogen.
in the primary structure, there was peptide bond is there. Hello, because amino
Ob-
acids they are held together with each other. Yes,
Obvio.
it was peptide bond because there is in sequence. All the amino acids they are held together by peptide bond. Got it? Secondary structure, another bond comes into action, which is hydrogen bond, both in alpha helix and in beta pleated. Correct? Hydrogen bond is there in the secondary structure. Correct?
Yes.
Now, tertiary structure, there is disulfide linkage or disulfide bridge. Okay?
Hmm.
So, what does it mean? Disulfide mean what? Two sulfurs.
Two.
Disulfide mean double sulfur. Okay? Two
Yeah.
sulfur. Now, if you remember, I told you one thing. Please remember one amino acid which has sulfur. What was that amino acid? Once I was
Cysteine.
teaching you, I told you one amino acid. You just remember it has double sulfur with it. It has sulfur
Cysteine.
in
Cysteine.
cysteine. So disulfide linkage will be made only between two cysteine amino acids. Wherever cysteine will come in front of cysteine, the bond will be made, disulfide linkage.
Okay.
Everybody will know disulfide linkage, but this thing, it is specific, okay? You have to know this thing because this is, I have
Okay.
seen this question in exams also. So cysteine, just wait for a while, okay? Cysteine will be giving this structure. Just hold on, okay?
Okay.
My battery is gone.
So can we continue the class on Thursday? Because I have my homework to do.
So you want that I should just give me 5 minutes to just skim through. Thursday, what is the benefit? You have the exam on Wednesday. So that is why I
Yes.
pre-pondered it. 5 or 10 minutes more we take. It will be, the class will be the same, like 1.5 hours you take. I will just skim through. You have class or you have to do some work.
Work.
Okay, then you know what we will do? I will be just skimming through with this thing because most of the thing I told you already.
Okay.
You know what? Because already we lost 5 or 10 minutes in between. Yes, in settling the class and everything. So those 10 minutes we will adjust here. What is the point? Otherwise, we will be leaving the class in between. So this is not good. Okay. This is
Okay.
a tertiary structure. It contains disulfide linkage and it contains hydrogen bonds. And second, there is one ionic interaction also. Ionic bond. Please remember, tertiary structure is containing how many three things are there? Hydrogen bond, disulfide bond, and third one is ionic bond. Okay, so it makes the structure more compact. Then what is quaternary structure? Quaternary structure is actually supercoiling. Same, mean tertiary structure is supercoiled again. You got my point? Hello?
Yes.
Yeah, so it will be coiled again, the quaternary structure. So here in the quaternary structure, only one thing that you will be adding, I will show it to you. You just add this point here, okay? Just you'll be adding this point, which is hydrophobic interactions. Hydrophobic interactions. This is hydrophobic interactions. Okay, now how many things are there? Hydrophobic interaction is there, ionic bonds are there in the quaternary structure, okay? Then disulfide linkages are there in hydrogen. So 4 bonds are there. So this is in the quaternary structure, correct? Then there is a general term.
So
There
in
is
tertiary structure also there are 3 bonds, hydrogen, disulfide and ionic.
yeah, disulfide and ionic. And quaternary structure, it has hydrogen, disulfide, ionic, and one more interaction which is known as hydrophobic interaction. This is supercoiling. Quaternary structure is known as supercoiling of the protein.
Okay.
Super. Acha, one more thing. There are 2 kinds of proteins already. There are fibrous proteins and there are globular proteins. You know this thing. I think we learned this thing in O levels. I taught it.
No, I do not remember.
You don't remember. So please remember there are 2 kinds of protein based upon their function. One are, I'll be doing it now. Because I'm recording it, so if you don't get it, so no problem. You can take it later on, okay? One is fibrous protein, fibrous, and second category is what? Globular. Yes. So fibrous protein, they make up the structure. Yes, make structure. Fiber. That is fiber. Yes, fiber structure. Collagen. Example is, for example, that is collagen. This is a fibrous protein. It make up the structure of your tissue or cell membrane, anything. Okay, so fibrous protein normally they have secondary structure. They are folding till the secondary structure. Okay, fibrous. But once we are talking about the globular protein, they can have tertiary and they can have quaternary structure, both tertiary and quaternary structure. Please spelling quaternary. They have both tertiary and quaternary structure. Globular protein, all enzymes, they have tertiary structure or quaternary structure. This you remember, okay? This structure which is shown in the picture, this is the picture of hemoglobin. So this is quaternary structure. This is quaternary structure, and this structure which is collagen, this is secondary structure where only hydrogen bonds are involved. You got my point? Yes. Okay. Last thing that is there, which you were talking about, there are 2 interactions which are very important: disulfide interactions or disulfide bond and ionic bonds. The bonds which are present, one is carbon bond, one is ionic bond, and one is disulfide bond. Disulfide bond is present always between what?
2 cysteine molecules.
2 cysteine molecules, correct? And ionic bonds are present in between carboxylic group and amine group because they are ions, correct?
Yeah.
Okay. Yeah. Once they are ionic, then you remember this thing. Once we are straightening our hair. Yes. With the dry
hydrogen bonds are
so there. Actually what we are breaking down, we are breaking down the ionic interactions. So this is our hairs, they get straight, but this is temporary with the heat or something, correct? Blow dryer, if we are using, so ionic interactions are removed. So that is why between the polypeptide chain, so structure becomes straightened. Okay. Yes. Disulfide linkage, once we are going for the perming or what we call rebounding, yes, where the permanent your hair become permanently straight. What exactly happening? You are breaking down the disulfide linkage. Once we break down the disulfide linkage, there is permanent change to the structure of the protein. Disulfide linkage is broken. Once it is broken, it is permanent. Once it is permanent, so once you straighten your hair permanently, so they remain permanently straight. If, for example, you have curly hair or I have curly hair, I straighten my hair, so those hair which got straightened, they will remain straightened. The new hair which will come down from down, they will be having curls. But the thing which we have already straightened, they will remain as such. Why? Because already we have damaged their disulfide linkages.
Yes.
I'm done. So I will be sending you this class also into, I will
Okay,
just
yeah.
record it, okay? And I will convert it and I will try to share it with you also.
Yeah, I also shared the book with you in the email.
You have shared the book
Yes.
in the
Can
mail?
you please check it? Yeah.
Okay, I will just check it. Thank you very much. Okay. Have a great
Okay.
day.
Allah Hafiz.
Allah Hafiz.